When Alexander Fleming noticed a stray mold destroying his bacterial cultures in 1928, he had accidentally isolated the first molecule capable of killing infection without harming human tissue. Within a few decades, massive assembly lines were pumping out these drugs by the ton. Bacterial infections that had killed humans for millennia, from strep throat to simple battlefield scrapes, were suddenly curable with a single dose. But bacteria reproduce at an astonishing rate. When we saturate their environment with lethal chemicals, any random mutation that allows a single bacterium to survive suddenly becomes a massive biological advantage. Over decades, this natural selection escalated. Strains developed resistance to multiple drugs, producing what we now call multidrug-resistant and extensively drug-resistant superbugs. Pathogens like MRSA and Acinetobacter baumannii can shrug off nearly every medication in our modern inventory. We weaponized nature to save millions of lives, and in doing so, we trained the exact organisms we were trying to destroy to become nearly invincible. Today, these drug-resistant infections kill more than 1 million people every year. The math is so concerning that the World Health Organization now ranks antimicrobial resistance as one of the top 3 threats to global human health. Compounding the problem is the fact that the pipeline for discovering new treatments has practically run dry. Scientists have already scoured the easy-to-reach places in nature and exhausted the obvious chemical combinations. Because developing these drugs is highly expensive and the financial returns are low, commercial pharmaceutical companies have largely abandoned the crucial early discovery phase of antibacterial research. If we continue to rely strictly on our older, failing drugs, routine surgeries and minor injuries will eventually become life-threatening again. Without entirely new methods of scientific discovery, we are rapidly sliding into a post-antibiotic era. To find new weapons, researchers are looking into what they call microbial dark matter. This refers to the roughly 99% of soil bacteria that simply refuse to grow in a traditional laboratory petri dish. Scientists bypass this limitation by inventing a device called the iChip. Instead of forcing bacteria into a lab, they place a diluted soil sample into this device's tiny chambers, wrap it in a semi-permeable membrane, and bury it back in the dirt so the microbes can grow in their natural environment. This chart and the accompanying scientific abstract details the discovery of a compound called teixobactin. It was the first major antibiotic found using the iChip, and it binds to bacterial targets with extreme precision. Unlike older drugs, teixobactin locks on to 2 separate targets at once: lipid 2, a precursor for the cell wall, and lipid 3. It effectively compromises the bacteria's structural integrity from 2 different angles simultaneously. Because it attacks 2 distinct lipid targets rather than a single mutating protein, pathogens have an incredibly difficult time evolving a defense against it. Other researchers are mining old data for missed opportunities. By deleting specific genes in known bacteria, they've uncovered hidden molecules, like premethylene myelinoasin C lactone, that were previously dismissed as mere synthetic intermediates, but are actually highly lethal to superbugs. By cultivating ungrowable soil and re-examining overlooked chemical stepping stones, science is finally tapping into deep biological reserves that were there all along. Some of the most dangerous pathogens, known as gram-negative bacteria, possess a brutal extra layer of armor. They have a dense, dual-layered outer membrane that actively repels or pumps out traditional small-molecule antibiotics. To breach this specific defense, scientists are testing a novel, experimental antibiotic called zosorabulbin. Zosorabulbin targets a specific protein complex called LPTB2FGC, the transport bridge gram-negative bacteria use to move building blocks to their surface. By blocking this transporter, the drug traps toxic molecules inside the cell. The building blocks rapidly back up, swelling the bacteria until it destroys itself. Global health organizations like GARP and biotechs like Antibio are now accelerating these specific hit-to-lead projects, focusing directly on the gram-negative pathogens prioritized by the WHO. We are moving away from carpet bombing the body with broad-spectrum antibiotics and shifting toward precise molecular sabotage of bacterial survival mechanisms. Instead of searching for new chemicals to kill these bugs, researchers at UC San Diego are developing ways to actively erase their resistance. they engineered a tool called the Pro-MOB-V system. It's a mobile genetic cassette built on CRISPR technology, programmed to seek out and delete the specific genes that allow bacteria to survive antibiotic attacks. Because these resistance genes live on small, independent DNA loops called plasmids, the CRISPR cassette can spread naturally through bacterial communities. It travels from one cell to another via conjugal transfer, a physical bridge bacteria use to share genetic material. Once inside, the CRISPR cassette identifies the sequences responsible for drug resistance and physically deletes them. This process renders the population susceptible once again to the very antibiotics they had evolved to ignore. By hijacking the microbe's own mating habits, genetic engineering allows us to actively disarm the evolutionary traits they've spent decades building. The 20th century relied on lucky, relatively simple chemical discoveries. Today, surviving the infections of tomorrow requires precision biological mining and direct genetic manipulation. Bacteria will always mutate. The evolutionary arms race between human medicine and microbial adaptation is a permanent condition of biology. Yet the invention of the iChip, the molecular sabotage of zocerabulpen, and deployment of CRISPR gene drives prove that humanity still possesses profound, untapped scientific depths to explore. The exhaustion of our old drugs guarantees a difficult transition, but by engineering entirely new ways to fight, we are proving that the post-antibiotic era is a complex challenge we are finally equipping ourselves to meet.